If you look around yourselves, you will see a lot of examples of living materials on every length scale that you can imagine. At the nanoscale, you have viral capsid, right? We look around, look at the animal kingdom, you can see different examples of functional materials generated by biology. And all of these are, through the principle of self-organization, and these are not 3D printed, they are grown, right? They oftentimes have amazing properties. I will just zoom in one example, like the butterfly wing. Turns out it has very, very fine structures, wing scales on this sort of butterfly wing, and they are made of course, chitin, like biocompatible, of course. And these structures actually can cause light to diffract and to interfere, generating different types of colors. And that could be useful for camouflage, thermoregulation, and also signaling between individual animals. As an engineer, I oftentimes wonder, how do we generate these types of functional structural materials from scratch? If you look through different examples, you will see primarily 3 pillars of living materials. They're oftentimes result of self-assembly, and also they're consisting of living and non-living components. And these different hybrid systems can have the sensory or actuating functions, depending on the specific context. So my lab, when we think about living functional materials, we are interested in addressing some of these fundamental questions. Along the line of generating self-assembly, we're interested in finding ways to program self-organized pattern formation using living cells. And in terms of the composite, we want to find the right molecules that allow the living cells to interface with their environment. And then we're also interested in finding ways to integrate different components to generate functional devices, right? Here's one example. This turns out to be a project that we have been working on for 2 decades. What's very interesting looking back is that when I was a postdoc, when I was looking for faculty position, you need to put together some proposed ideas. Looking back, the programming pattern formation is actually the only project that I'm still working on. So it has been a long going, right? And this work actually was pioneered by 2 former grad students in the lab, right? Initially, we tried to build the classical Turing circuit. The basic concept of the Turing circuit is actually very intuitive. The notion is that you have 2 molecules. One is an activator, one is an inhibitor, right? Activator activates its own synthesis and also drive the synthesis of the inhibitor. And the inhibitor can come back and can suppress the production of the activator. Now, if the inhibitor diffuses faster than the activator by a certain ratio, you could generate the so-called Turing instability. If you start from homogeneous initial conditions, you can generate well-defined spatial patterns. That was our initial motivation. So we came up with a design where we have a transcriptional factor, essentially, T7 RNA polymerase, driving its own synthesis. And then it drives the synthesis of a small diffusible chemical, acetylhomoseronactone, which at high enough concentration can lead to the expression of a repressor that suppresses a positive feedback. So this part is analogous to the Turing circuit, But on the other hand, there's a fundamental difference. In this case, the activator does not freely diffuse. So it's moving around, being carried around by cells. So as a result, we're not generating Turing patterns. Nevertheless, this circuit actually can generate very nice self-organized pattern formation, right? Here's an example which was actually made by another former grad student, the self-organized ring pattern formation by the circuit. You will see the cells growing, right, on the surface of the agar. And what you will see next is the formation of a core, somewhat red, which is indicating the expression of the repressor lysozyme. Then you'll see the colony growth stopping, in part due to the exhaustion of the nutrient. And then you'll see the emergence of the ring near the edge of the colony, which matures towards the center. So all of this is self-organized. So we don't have a pre-patterns for the colony to follow. The cells are talking to each other, figuring out where they are, then they control their gene expression, leading to the expression at the right location and right time, generating the self-organized pattern formation. What's also interesting is that this pattern actually has this property of scale invariance. The idea is that if you control the growth environment, for example, the overall size of the growth domain, if you provide more space, the colony will become larger. At the same time, the patterns will become thicker. What's really fascinating is that the size of the pattern scales proportionally to the colony size. So they obey this scale invariance. So this is very much analogous to the scale invariance we will see in animal kingdoms. Like different humans, even though we come with different heights, the proportion of our, say, arms to our body size is roughly a constant. So it turns out this simple circuit actually can exhibit some property of the scale invariance. By the way, this was actually, we got a lot of advice from Chris Rao at University of Illinois, right? It was wonderful collaboration. On the one hand, you can use this sort of self-organized pattern formation circuit as a way to probe some open biological questions. On the other hand, if you have the ability to generate self-organized pattern formation, you can use this as a foundation to build up the complexity and generate essentially structured materials by using the pattern forming bacteria as the scaffold, right? So in this case, what we have done here is actually very simple. We have this pattern formation circuit, right? On top of that, we introduce a recombinant protein, which we got from Tim Nu's lab from MIT. So they engineered this protein, which can allow the living cells to bind to gold nanoparticles. So what we did is that we actually conduct the pattern formation experiment in 3D now. And in 3D, this circuit actually can generate this self-organized dome structure. And now, because these cells carry this recombinant protein, now we use this protein to interface with inorganic materials. So use the dome structure to assemble the gold nanoparticles into this structure. In essence, we're using the self-organized patterns generated by the bacteria as a scaffold to assemble structured materials. And if you have two of these structures being assembled, you put them opposing to each other, gold nanoparticles are conductive. If they're separate, you don't have a current, but if you press them together, you have a current. And if you press harder, the conductivity is greater, so you have greater current. So this can serve as a very basic pressure sensor. And this was demonstrated by the former grad student, right? And so here is the bacterial fabric structure is enclosed in here, right? We form a, if you press these two structures together, you have form a closed circuit. You can, light up LED light. And if you track individual frames, you will see that the intensity of the light actually correlates with the strength of the pressing. So we did a lot of the calibration. This actually serves as a rudimentary pressure sensor. Of course, you may not necessarily care about pressure sensing, and this by no means is a perfect pressure sensor. But the key point is that if you're, I'm sure you got this, this is a Morse code for Duke. But the key point where we try to convey is that once you have ability to generate self-organized patterns, you can actually use this as a foundation to assemble structured materials with well-defined properties, right? This brings up another point. We actually don't see many examples in synthetic biology, in terms of being able to generate self-organized patterns. I'll touch upon that point later, a little bit more later on as well. I see two potential limitations. One is that this is like a feedback. On the one hand, we need certain degree of technical capability. On the other hand, we have to convince ourselves that when we have the ability to generate the patterns, they are useful for something, right? So there are these two aspects are tied to each other. So far, we don't have many applications, in part because we don't have sufficient capability. And because we don't have enough applications, that actually reduces the demand for us to push the technical capability. So I would argue, right, a lot of the applications in synthetic biology or in pattern formation could be inspired if we develop greater degree of capability that will inspire new additional applications. So what we want is this scenario. We have much greater technical capability, and that will inspire more diverse applications. And when we have more applications, that will impose more demand for us to improve the technical capability. And this has been slow in terms of programming pattern formation, unlike some other aspects of synthetic biology, right? For instance, if we look back of the 20 years of the modern synthetic biology, there has been tremendous progress. I would argue the vast majority of the efforts have been focused on generating logic gates. As with increasing difficulty and with decreasing number of examples, we're also generating more sophisticated dynamics like switches, bistable switches, or ultra-sensitive switches, or oscillations, right? In contrast, there has been a profound lack of progress in programming self-organized patterns from scratch. Every time I do this search, this is a very crude estimate. I did the PubMed search, just type in the keyword of synthetic biology. Every time I do this search, there is a huge bump in the number of publications, right? As it stands, it's more than 13,000 publications on synthetic biology. There are fewer than 10 examples of engineered gene circuits able to generate self-organized pattern formation, right? Then the question is why it is so difficult to program self-organized pattern formation. So this is both a computational problem and experimental problem. Here are the three challenges in our experience that we feel like these are kind of represent bottlenecks. One is that lack of quantitative understanding of the components. For example, as I mentioned earlier, when you program pattern formation, you need to understand the dynamic range of the regulatory functions. You need to understand, have some good estimate of how fast the different components actually move around, and specifically how they move around, right? By what mechanism, by what biophysical mechanism, if you want to design from the first principles. And even if you have the perfect circuit, using the simulation to probe the circuit dynamics is also a very difficult problem. It's difficult in two ways. One is that if you do numeric simulation, it's much more time consuming than the models of some other dynamics, right? If which can be described by algebraic equations or by ordinary differential equations. One way to think about spatial dynamics is that you can, of course, you can use agent-based simulations, which is most time-consuming. Or if you use do some coarse graining, you can have partial differential equations. In comparison to ODEs, PDEs are essentially solved as a series of parallel ODEs, right? Depending on how the resolution of discretization in space. So it's much, much more time consuming to solve these sort of models, even if you had the perfect circuit, right? And of course, these are also more difficult experiments to do than, for example, than checking whether the circuit is on or off, right? And these are the fundamental bottlenecks, limiting the progress in programming pattern formation. So our approach is increasingly taking these two strategies. So how do we do more effective coarse graining? And how do we utilize the advances or the tools in machine learning to accelerate our predictions and make more accurate predictions, right? I'm going back to the circuit I just described to you. And initial model was done using an agent-based model. Later on, we developed a PDE model that performs equally well. So it's much, much faster. It was about 100 times faster than the original agent-based model. And so here, I'm not going into details of this PDE model, but emphasize what we're generating. So here is a readout of this circuit. So what we're generating is after a certain time window, we look at how the expression of the system output here is reading out M cherry as a function of the location. So in this case, you will have high expression in the center and high expression near the edge. So you have this scoring pattern. So x-axis is the position, Keep in mind, every simulation here, in this case, so I'm looking at the chat. Yeah, feel free to stop me. Either type the question in the chat or just unmute yourself. Do the self-organized assemblies contribute to biofilms on glass or a larger material? Yes, I would say in general, you could consider the biofilm formation as part of the self-organized pattern formation conducted by nature, right? They oftentimes generate incredibly fascinating structures and patterns. Our ability to even to explain them is still very limited. I actually give some more examples on that later on. I hope that answers your question. Right. So when we do this simulation, of course, there's always room to improve the efficiency one way or another. But just as a ballpark, this simulation takes about 5 minutes per parameter combination. And that's a very important parameter to keep in mind. Now, in ongoing research, we're thinking about two complementary approaches. If I want to better understand in specific examples the fundamental insight leading to the pattern formation process, one approach we're taking is to do more extreme coarse graining. We can, given some constraints, we actually can formulate this patterning process by a set of ordinary differential equations, which will be more intuitive, will be much, much faster to solve. The other, of course, the limitation is that it's more limited scope because of this more stringent assumptions. In the other extreme, we're taking a data-driven approach where if we don't care about the mechanism at the get-go, then we just want to have fast and accurate predictions. And we are using machine learning to complement the mechanistic models. Well, downside is that by itself, these predictions are difficult to interpret. In our case, however, we do have the reasonably mechanistic model. So when we have a prediction, we can always check why a certain prediction behaved in a certain way, right? So this is just one slide summarizing several years of work, actually in collaboration with team, right? So the basic concept here, I will only explain the basic concept, is that there's a class of the colony growth dynamics that satisfy these sort of constraints. It's approximately 2-dimensional, and they have the radial symmetry, and they're closely packed. So for this type of colony growth and pattern formation, and we can actually track the trajectory of a representative cell being pushed by the force generated by cell growth. And with proper formulation, you can actually describe this process by simply a set of ordinary differential equations, which is much, much easier to analyze and to compute. And in this paper, that we actually demonstrate the prediction of the colony growth and coupled gene expression dynamics, which is consistent with our TDE model. And the foundation of this colony growth modeled by the set of ODEs, we actually did thorough validation using Tin-Lu's agent-based simulation, right? And actually, the simple model captures the qualitative aspects of the colony growth and expansion, even if some of the basic assumptions are violated. For example, the cells don't have to be spheres. Even if you have other shapes, this model performs very well. So I want to spend more time talking about approach that's we're adopting in a number of projects. Coming back to the point of the PDE simulation, it takes about 5 minutes per simulation, which by itself is not a big deal. But now, if I have this model, let's imagine I have 10 parameters, and I want to explore 6 values per parameter. Then I need to conduct this number of simulations, and that is a big deal. We estimate it takes about 600 years to numerically explore the parametric space. The key point I want to make here is that even if I had the perfect model, I may not know the full capacity of the circuit, even by numeric simulations, which apparently seems to be much faster. It's 5 minutes per simulation, right? So what we decide to do is saying, well, maybe we can benefit from using a neural network. So the concept is very simple. What we care about is that for different combination of parameters, we want to check the final outcome, whether that's interesting to us, even before trying to understand why the outcome comes about in that way, right? We may not necessarily care about the mechanism at the get-go. We care about them later. So we just want to ask the question, does the circuit have the ability to generate something interesting? So we want to use a fast computation to screen the parameter combinations to identify something that's interesting, right? So what we did is saying we use the mechanistic model to generate a large enough number of simulations, which is sufficient to train a neural network reliably well, but not so large such that even the generating the training data becomes computationally prohibitive. Then we use this data generated by the mechanistic model to train a neural network. And then we use the neural network to directly establish the mapping between the parameters and the predictions. So that's a very, very simple concept, very, very simple concept. It works extremely well. It can achieve 30,000-fold acceleration in computational speed. Of course, this in a way is like cheating, right? So because when I'm solving the PDEs, it requires sufficient resolution in discretizing time and space, which is time consuming. That's why it's slow, right? Well, when I was making predictions using the neural network, I actually didn't care about the intermediate steps. I only care what the final outcome I can generate. So the neural network is bypassing all of these time-consuming processes. That's why we're getting tremendous acceleration in the predictions. And it performs extremely well, right? Here are just some examples. On the bottom, you'll see the simulations from the PD model. On the top, you see the corresponding neural network predictions. By and large, the neural network does a very good job in matching what the PD is predicting, right? Occasionally, the neural network is saying ghost. There's nothing going on according to the PD model, but the neural network is saying all sorts of fancy stuff, right? Which it clearly is wrong. So how do I deal with this situation? Of course, you don't want to validate every prediction one by one. If you were to do that, you're defeating the purpose of using the neural network, right? So what we decide to do is to use a very simple strategy, is to do ensemble prediction. So the notion is that given the same parameter set, I should say the training set, I will train 4, it doesn't have to be 4, just multiple neural networks independently. Of course, the training process has intrinsic randomness, depending on how you initialize different neural networks, right? And when I do the training, right, every neural network can make its own prediction. And then we just check the consistency, like the voting result. So in this case, like the majority is predicting this, I can use this as a ensemble prediction. More critically, whether these predictions are consistent or not consistent, or give me a metric on how confident the predictions are. So we actually check that, right? The idea is here, this is a disagreement between predictions made by the neural networks, how much they disagree with each other. And this y-axis is a difference between the neural network predictions from the ground truth, which is simulated by the mechanistic model. You see a very strong positive correlation. So the basic idea is that if the neural networks agree with each other, it's more likely to be a reliable prediction. But if the neural networks disagree with each other, then it's less likely to be reliable. So you, as if you are doing these predictions, you get to set where you want to have high confidence in the prediction, right? Like in this quarter, it's very, very accurate. For most of the examples, this is actually, I realize this corresponds to that one. This corresponds to the bottom one, right? So this provides an intrinsic metric for us to evaluate how reliable the predictions are. To recap, so this is a very basic, a simple concept, which is that neural networks can enable massive acceleration in predictions. You can always check the to what extent the neural network predictions make sense by actually looking at the corresponding mechanism-based models. And votings actually enable self-contained evaluation of the quality of the prediction. We also demonstrate that this is generally applicable for some other models, including stochastic models. You want to use in a context where the simulations are very time-consuming, such that you can gain. But on the other hand, it's not so time-consuming. You won't even be able to generate sufficient training data. And the latter is actually quite a challenge for some of the examples we're trying to tackle. Now, coming back to the challenging programming pattern formation. To date, I mentioned there are only a limited number of examples of self-organized pattern formation, including a pioneering work from Chenli Liu, who is now in Shenzhen Institute of Advanced Technology, the sequential ring pattern formation. So this is one I just described briefly. And also a wonderful example, Ting and is involved in, right? And Dave Correct, and both of whom I know very well, coming out of Ron Weiss's lab, right? stochastic Turing pattern. If I look at all of these patterns, they're relatively simple. In comparison to nature, our ability to generate more complex patterns indeed is very, very limited. Here's an example. This is just simply a picture taken by microscopy picture taken by a current postdoc, Lan No, right? And it's very, very beautiful. This is generated by a model organism called Pseudomonas aeruginosa. It's an opportunistic pathogen, but here we're only focusing on their pattern formation capability. It actually won the top prize of the Pratt photo contest a couple of years ago. And now it's on display in the new engineering building. And this was taken by a larger grad student of this photo on display. It's gratifying to see that people actually appreciate the beauty in the scientific data. This is what we try to understand. So the question we're asking is, how do we model and control and use these sort of branching patterns? I mentioned that the patterns we can program are relatively simple, that limit the possibilities of different applications we can think of. So the somewhat basic question we're asking is that if I have the ability to generate much more complex patterns, then maybe I can think of more diverse and more interesting applications going beyond the pressure sensor, right? This turns out to be a very, very difficult problem, much more difficult than we initially anticipated. We're not the first to look at the branching patterns. It has been looked at for many years. And the typical approach is to build a biophysical model or reaction diffusion model. I'm not going into details of this, to look at the conditions where you lead to the emergence of the patterns, right? And here is like we generate this model that generate the emergence of the patterns. In that regard, it's good. It's consistent what people have generated in the literature. But quantitatively speaking, we're missing out of some very important features of the patterns we see in the experiment, right? And the quantitative agreement is not great. For instance, in the experiment, we tend to see the branches can be very, very thin, right? Along each branch, the width tend to be approximately constant, right? And we tried many different formulations. It's incredibly, I would say, depressing. It's even depressing to me, right? Multiple years. And what's most depressing is that when we look at this sort of modeling effort, when we see a disagreement that we cannot feel, we actually don't know what's wrong. We don't know if the discrepancy is due to the model missing some critical components, or, for instance, I could write down one equation formulation not in the right form, then there's no hope, right? Or even if the model is in the right form, there may be parameter space we simply technically cannot fully explore. We're not in the right parameter domain. We actually don't know which case this corresponds to, right? Because every simulation is very, very slow, very slow. Instead, we want to take a step back, ask the question, can we find a larger way of describing the patterning process that will enable us to make reliable and biologically relevant predictions? So the approach we're taking is more extreme coarse graining. We ask the basic question, let's assume the branches can emerge. We don't worry about how, right? We just assume they can emerge. Based on the observation, right, these branches are typically constant width, right? We assume every pattern just characterized by two parameters, branching density and branching width. Of course, when the cells grow, when the colonies become larger, there will be additional branching and to maintain certain degree of branching density. By branching density, I mean how many branches you have per unit length, right? So this paper is actually almost accepted. I think the reviewers really like it. And these are pending some minor edits in the text, right? So this is kind of an advertisement of a paper to come out. So if we take that approach, turns out we can use a very simple model where we still model the nutrient as like using the reaction diffusion equation, where this is consumption, this is diffusion, right? For the other components, we can model actually use ordinary differential equations. Ordinary differential equations. You have the growth and have the extension, right? Then we ask, using this simplified formulation, before I go into that, so this is a sample simulation. I want to emphasize this is orders of magnitude faster than the PDE model. I actually generate characteristics which agree much, much better with the experimental data, So using this model, we're just asking a basic question. How does the overall system or the overall system output, in addition to patterning, how the overall biomass depends on these two parameters? Turns out in each growth environment, so in our simulation, that's set by the amount of nutrient that's available. The colony biomass accumulation depends on branch width and density. Showing here is actually the patterning process. If I increase the branching density, you see much, much denser branches, all right? If I increase the width, then you have thicker and thicker branches. Above this line is when these branches start to overlap with each other. You don't have any branching anymore. It's just a circular colony, right? And we can quantify with higher resolution of the biomass as a function of these two parameters. What's interesting is that for a given environment, you will actually have optimal biomass accumulation for some sort of intermediate width and density. Then we ask, to what extent this optimal growth will depend on different growth environments. So how does this shift according to different growth environments? Indeed, that's tunable outcome. So the optimal branching densities are tuned by the growth conditions. There are two ways we can tune the patterns and the growth. One is controlling the amount of the nutrient. One is to control how fast the cells can move around, which influence the branching, the colony branch extension speed, right? What the key point I want you to focus on is the yellow portion that shifts according to the nutrient environment. When I have increased nutrient while fixing the motility, I will see increasingly the optimal patterns to have increasingly denser and wider branches, right? And if I increase the motility of the cells, for example, by reducing the agar density, I will tend to have much more dense branches. So these are all shaped by the growth conditions. These predictions are actually consistent with how Pseudomonas respond to these growth perturbations. So in other words, Pseudomonas appear to approximately follow this optimization rule, that if I increase the nutrient concentration, this is increasing the decrease in agar density, right? Decreasing the agar density, so increasing the colony expansion efficiency. So these are the numeric predictions. And these are the actually experiments. There are much more extensive experimental data to validate the consistency between the modeling and experiment. So the key point is that the Pseudomonas appears to approximately follow this optimization rule. Now, given that, we can actually take advantage of this property and make predictions of the pattern formation of Pseudomonas under different conditions. One example is actually very intriguing, is that Pseudomonas can have this anti-nutrient gradient expansion, right? This is a prediction. If you have, in this case, you have a gradient, there's a higher concentration of nutrient on this side, lower nutrient concentration on the left-hand side, the branching actually extends more on the left-hand side, right? And this is experimental data. In the experiment, this is how you can create a nutrient gradient by generating the agars, one containing the nutrient, and then you pour the agar without the nutrient. So you can create this nutrient gradient. This turns out to be non-trivial behavior of the system that the model actually can capture. Of course, you can predict more sophisticated scenarios. And these are the predictions of how the branching pattern gonna respond to the growth environment. You can think of this as another way of controlling the patterning process. Or you can predict the colony patterns with various initial seeding configurations. So in every case, the model is doing a terrific job in terms of predicting the outcome that's consistent with experimental observations. Well, there's more. There's just very beautiful pictures, right? And these are the initial seeding patterns, and these are the model predictions, and these are the actual experiments. Well, now coming back to the point, now we're developing the capability of predicting and explaining these sort of patterns. So how do we want to use these patterns? We have ongoing research. Now, if we have this ability to generate these sort of patterns computationally and experimentally, one question is essentially using biology for us to compute. The notion is that the pattern generation process can be considered as encoding process. If you have certain initial configuration, you generate the phylo pattern. That's information encoding. Now, starting from this side, if I can deduce my initial configuration, then that's a decoding process. So in the ongoing work, we're trying to explore that to what extent you can utilize self-organized pattern formation as a way to generate this information encoding and decoding in a robust manner. Another aspect we're interested in collaboration with some of our colleagues in the materials engineering, is that if I can control this sort of branching pattern process, if we introduce the right effectors, then can we generate this sort of branched materials, which could have optimized transport properties for different applications, right? These are the potential applications we're currently exploring. Now, coming back to the notion of living functional materials, let me check if I have 10 more minutes. Is that right?
Yeah.
Okay, thanks. That should be sufficient. The other aspect we're exploring is how do we integrate and generate actually functional devices by combining engineered cells and with stimulus responsive materials. So this aligns line of a platform we're engineering, which we call Microbial swarmbots. It's a loose term. The key point is to couple spatial segregation and environmental sensing to generate well-defined functional devices, right? So the idea is that we have engineered bacteria with certain types of sensing and actuating capability. We encapsulate groups of these bacteria, and this provide a well-defined interface for the group of bacteria to carry out their function. So we'll want to realize collective sensing or actuating functions. The proof of principle was done a few years back, right? Demonstrating how you can program well-defined functions in this format. And this is like the second generation where we engineer the bacteria, they can sense the spatial confinement. We also chose a material that can respond to bacterial growth and death. In this case, it's a very common and biocompatible material, chitosan, right? And here's a basic operation of this device. You have engineered suicidal bacteria that they can pop open at high enough density, and we encapsulate them. They will grow, right? As they grow, they are going to change the chemical environment. For example, changing the pH and ionic strength. So they, in response to this changing chemical environment, what's really interesting, this is just the property of the chitosan. The chitosan will shrink. And this shrinking process turns out roughly coincides with the popping of the cells. So in essence, you create a hybrid device where the cells grow and they're gonna pop and the material gonna shrink and squeezing out the content produced by the cells. When the cells pop, they release their protein content. And you can use this as a integrated separation device, right? You can reset this process, keep going, and produce and fabricate proteins. So here is a basic building block. This was a circuit we built a few years back. This circuit logic is very simple. We call it EPOP. You have high enough cell density will lead to higher copy number of the plasmid, leading to higher gene expression of a toxin, E protein from 5X174, which blocks the cell wall synthesis, right? So as a result, you will see the population level oscillations due to this negative feedback control, right? Now, if you integrate with the SwarmBot platform, now this is the microfluidic device we use to quantify this response. This SwarmBot plate here, right? This is where we put in the nutrient, and downstream we can do dynamic analysis, protein purification, and pathway assembly. So here is Microbial SwarmBot in action. So the nutrient flow in periodically. You see every time you flow in, it's resetting the size of the swarmbots, right? And you can do downstream analysis and protein purification, etc. Right? Here's one example. Well, here is just a brief explanation how we can couple this device with downstream processing to generate, to enable versatile protein production and purification, right? A larger aspect we're working on right now, which I find particularly exciting as well, is the notion of using this as a platform to realize division of labor. The notion is that if I have one type of swarmbot producing one protein, another type of swarmbot producing another protein, and you can control the stoichiometry of these two proteins simply by controlling the ratio of these two types of swarmbots. You can generate different types of protein complexes or tune the overall output of a metabolic pathway, right? We actually demonstrated the latter as a proof of principle. We chose a well-characterized metabolic pathway just as a model system. This pathway requires multiple enzymes, right? Multiple enzymes. It's not important to remember what of these enzymes are. It just requires multiple steps. That's a key point to keep in mind. We generate multiple swarmbots, each producing and releasing one enzyme, right? And then we put all of them together and generate one part culture and then do the purification. Then we can directly generate the enzyme mixture. And turns out this can directly reconstitute the metabolic pathway and generate the desired product. So this is a proof of principle of demonstrating using the microbial swarmbots to do optimize, to reconstitute and maybe optimize metabolic pathway activities. Here we're directly using the enzyme mixture. In ongoing research, what we're interested in is actually directly use the microbial swarmbots to control the metabolic pathway activities without doing the purification. So this, I'm going back to this essentially the summary slide. I give you a few examples how we think about programming living materials, addressing both of the fundamental bottleneck and also demonstrating some potential applications, right? One aspect of research I didn't have time to go through is facing a similar computational design challenge of high dimensionality. is the dynamics of microbial communities. And the basic question we're trying to understand is, given a particular microbial community, how does it respond to different types of perturbations? And under this umbrella, we're interested in gene transfer dynamics, screening drug responses, and thinking about intervention strategies, and also basic questions of ecology and evolution, right? With that, I would like to thank the people who actually did the work. In this particular case, I had the pattern formation project, was pioneered by a few former grad students, right? Primarily, the work I highlight is Will Cao, and then former lab member Sangying Wang, and a current postdoc Lan and grad student Jia Lu, and the funding agencies. With that, I'm happy to take any other questions.
Thank you, Lingchong, for the very inspiring talk. I enjoy very much. So it looks like we have a question right now in the chat.
So how does the seed Pseudomonas work with the two plasmids to generate others with a second ring and so on? Oh, that's a fascinating question. We're still trying to understand. We'll explain the branching patterns by the Pseudomonas by themselves. We indeed are interested in how, if I introduce the ring pattern formation circuit into Pseudomonas, that was actually initial motivation. But explaining the Pseudomonas branching pattern formation turns out to be a harder problem than we anticipated. So we actually spent last several years just trying to understand the chassis a little bit more. I hope that clarifies your question.
I have a question. This is Sergey Maslov.
Yes.
Thank you for a wonderful talk, Lingchong. My question is about the part of the talk where you use neural networks to predict the outcome, the spatial profile of your simulations. So I can imagine that the parameter dependence is characterized by a fairly complex phase diagram where you may have some discontinuous transitions. Is it possible that some of the cases where you had a large error between the predictions and the actual outcome are somewhere near those phase boundaries? And overall, whether you try to use your technique to actually just plot the phase diagram of your complex system of equations.
That was a very, very insightful comment. I very much agree with your intuition. If you look at our discussion, that's precisely what we wrote, our speculation. We don't know for sure. The notion is that I suspect that's indeed the case, where if you have system sensitivity being very, very high, for whatever reason, if you have discontinuity, right, that's where the neural network can potentially fail, right? In a way, we have been thinking about maybe we can use the failure of the neural network to map out the sensitivity landscape of the dynamical systems. That's something we have been thinking about. We made some progress, but we don't have definitive conclusions yet. But that's a very insightful comment. Yeah, that's what we wrote in the paper, actually.
Thank you.
Lingchong, I have a question too. So it's a very general question. So apparently, pattern emerging from native living systems, such as like Pseudomonas showed in your example, is often very robust and elegant, while those from synthetic strain through the introduction of gene circuitry are often noisy and fragile. So assume that those synthetic strain may miss some like key trait, which may differentiate like the behavior of engineered strain versus native strain. I'm wondering, do you happen to have any insight about that? What we are missing toward the creation of robust gene circuitry that give rise to like such a stable self-assembled structures?
Yeah, that's a fantastic question. This is something we're still learning. If I understand you correctly, you're saying that let's say Pseudomonas can form these sort of branching patterns, which are super reliable, right? Reproducible.
Yeah.
That's indeed the case. They are fragile in a different way. This is actually some result we're wrapping up a paper on. I can just mention very briefly. It turns out they're robust only if you always start from the liquid culture that's stored in the freezer, right? Now, if you propagate Pseudomonas on solid phase, that feature of programming branching patterns can quickly change. It's very prone to cheaters, right? And that's one thing we are trying to better understand. So whether they're robust or fragile actually depends on the specific experimental protocols we're taking. We're not yet sure if these sort of patterns are more or less robust than some of the synthetic circuits we're building. But it's not necessarily a fair comparison. For instance, in the ring pattern formation, it's quite robust, quite reproducible, but then it's also simpler. It's simpler, right? It's easier to reproduce that features. For the branching patterns, it turns out to be quite dependent on how we grow the cells. I actually don't know the answer to your question. In short, we're still learning.
Lingchong, have you tried, and what are some of the challenges in trying to grow 3D patterns?
We tried a little bit, but we haven't tried hard enough. The little bit we tried is the example I mentioned, the Nature Biotech paper. We simply grow the cells on top of a membrane, which allows us to control the nutrient flow, and also make it easier for us to do downstream processing. And in that case, if we don't impose a constraint on top, we do see the 3D colony growth and leading to the formation of the dome structure. But that's the extent that we have tried. Indeed, I think it's actually fascinating to think about how we can generate 3D patterns. That will bring up an even more difficult computational problem.
Right.
The two-dimensional simulation is already causing tremendous computational challenge for us. If I want to simulate in 3D, if I use PDE model or something agent-based model, that I can see a fundamental challenge there as well. But it's really fascinating. That should be something we should, as a community, we should think more about how we predictably control two-dimensional or three-dimensional patterns.
Thank you.